Okay, so today's topic is Enterobacteriaceae. It is from the subject veterinary microbiology, and this is one of the most important families of bacteria. So this is Enterobacteriaceae general features. After that, E. coli part 1, and then we will make E. coli part 2 in a separate video. So let's start. So Enterobacteriaceae, it is a gram-negative bacteria, bacilli, that is length 3 micrometer. Bacilli that is rod-shaped, usually ferment glucose, but E. coli ferments lactose. Important point. It is catalase positive, oxidase negative, just like staphylococci. The motile enterobacteria, any bacteria which is motile is because of flagella. Any bacteria which is motile is because of flagella. But this Enterobacteriaceae is motile because of peritragus flagella. Peritragus flagella, this is the diagram for peritragus flagella. These are the flagella present all over the cellular surface. So this is called peritragus condition. We'll check that later. So major animal pathogens are E. coli, Salmonella, Yersinia, which you have to study. Okay, these are the bacteria examples. Okay, now usual habitat. Usual habitat inhabit the intestinal tract of animals and humans. So these are commensals in the intestinal tract of animals and humans. But when their population extremely increases, so that leads to infection, either intestinal or extraintestinal infection. Now differentiation of Enterobacteriaceae. So any bacteria which wants to be placed in the family Enterobacteriaceae has to follow certain characteristics. Okay. So these are the specific features which are specific to Enterobacteriaceae. So any bacteria which wants to be get placed in the genus Enterobacteriaceae, in the family Enterobacteriaceae, has to fulfill these 5 conditions. So it should be catalase positive, oxidase negative. It should grow on Mekon-ki agar, most important characteristic. It should be gram negative, bacilli, and facultative anaerobe. Facultative anaerobe means there are 3 conditions: anaerobic, aerobic, facultative anaerobe, and obligate anaerobe. So aerobic, that is, can grow only in the presence of oxygen. Facultative anaerobe means that they can grow in the presence of oxygen also, but their most preferable environment is in the absence of oxygen. And obligate anaerobe will die in the presence of oxygen. You can only survive in the absence of oxygen. Okay, so that is the case. Now Enterobacteriaceae are of 2 types: lactose fermenter and non-lactose fermenter. So lactose fermenter, their colonies and surrounding medium are pink due to acid production from lactose. Okay, and non-lactose fermenter, colonies and surrounding medium are pale. Because why? Because alkaline due to peptone utilization, and alkaline due to peptone utilization. So these are not pink non-lactose fermenter. So lactose fermenter example is E. coli, but non-lactose fermenter examples are various Salmonella. So okay, now colonial morphology. So various different examples of Enterobacteriaceae show different colonial morphology. External structure of colony. So mucoid colony is shown by Klebsiella and Enterobacter. Swarming, that is groups. Groups are made by Proteus, and red pigment production is done by Serratia marcescens. So these examples are important. Also, this data is also been taken from Quinn and Carter. By the way, all these notes are from Quinn and Carter. So if you like them, please subscribe. So virus is measured in nanometer, bacteria, dimension of bacteria measured in micrometer, and bacterial and fungal colony measured in millimeter. So these are important. This question was asked in our first midterm in second year microbiology, that what is the unit for measuring the dimensions of bacteria. So it is micrometer. Mine was correct, but many did wrong. Many wrote nanometer. So that is for virus only. Okay, next is Escherichia coli. So this is a part 1 for Escherichia coli. So Escherichia coli, it was first isolated or discovered by Theodore Escherich. That is why it is given the name. This is E, not I. Escherich. So that is why it is given the name Escherichia. Okay, that is why it is given the name Escherichia. Strains of E. coli produce metallic sheen, green metallic sheen on EMB agar. This is the characteristic feature of green metallic sheen. Means the green color, the colony will appear green, shiny green, but this is the characteristic feature of E. coli, not a differentiating feature. Another bacteria also produces green metallic sheen. Okay. Now types of antigens of E. coli. So there are 4 types of antigens: O, K, H, F. So if we talk about O antigen, it is a somatic antigen. This is LPS in nature, lipopolysaccharide, and located at the surface of cell wall. This is the O antigen, somatic antigen. Next is K, capsular antigen. This is capsule, so capsule antigen K, polysaccharide in nature. H and F, fimbrial and flagellar and fimbrial adhesion respectively, protein in nature. Okay. Okay. Now what is the source of transmission of E. coli? This is mainly a topic for this source of transmission is mainly a topic for patho, but we will discuss it here since it is given in Quinn and Carter. So source of transmission is ingestion of contaminated feed and water first, and second is direct contact with infected animals. Okay. Now there are two types of strains of E. coli. General, arbitrarily, there is no scientific reason behind this, but this is arbitrarily divided. This classification of E. coli is arbitrary, just based on convenience. So first is causing enteric infection, second causing extraintestinal infection. Enteric means intestine. Okay, easy? Okay. Now this point is very important. E. coli 0157H7 is a major foodborne zoonotic pathogen in humans. Very important. We have to learn this point. Okay. Now pathogenesis and pathogenicity. So what is pathogenesis? Pathogenesis is defined as a progressive development of disease, and pathogenicity the ability of an organism to cause disease. Okay. Now virulence factor of E. coli, there are various virulence factors with the help of which it causes the diseases. Virulence factor are the factors which have the function of causing damage to the host tissues or killing the host tissues. So capsule, endotoxin, enterotoxin, structure responsible for adherence and colonization. So these four are the main virulence factors of E. coli. So first is capsular polysaccharide. So capsular polysaccharide interfere with phagocytic uptake of E. coli. Okay. So what is the meaning of this? That is capsular polysaccharide or capsule. What it does is it do not let the phagocytes of the animal's immune system to phagocytize the E. coli, to engulf the E. coli. Okay. Capsule is weakly antigenic. Weakly antigenic means it does not lead to mass production of antibodies. Now capsule also interfere with the complement system. So what is complement system? Complement system is a part of immune system which helps the immune system to specifically pick up the foreign particles and kill them. Okay. Okay. So this is part 1 for E. coli. Follow for more.